Now I'm going to shift attention from design and data collection to monitoring. In the mid-1960s, the NIH, in particular the National Heart, Lung, and Blood Institute, set up a structure as to how trials should be conducted. And more or less, this structure has carried forward in time. On this diagram, I've illustrated some of the key components. We need a funding agency, someone who is supporting and paying the bills and providing some overall guidance. We need a steering committee, those who are going to write the protocol and manage the conduct and see to it that everything is going according to plan. This steering committee may be made up of members from the academic community and from the funding agency or the sponsor. And someone is usually identified as being the chair of the steering committee. We have various working components. Committees that define eligibility criteria, assess outcomes, central laboratories, clinical laboratories or EKG laboratories or X-ray laboratories. We have the clinics, of course, where the patients may be, or at least recruited through. A data coordinating center where the data are collected, quality controlled and analyzed, and then a monitoring committee, which is monitoring for safety along the way. The Data Monitoring Committee has gotten recent attention. In the year 2000, because of a few deaths in some gene transfer studies, the Department of Health and Human Services wrote an article and issued a mandate that all NIH and FDA trials must have a monitoring plan. And for some trials, a separate special monitoring committee might be required. The NIH in 1998 had been working on such a plan, and this was published, made available, that all sponsored trials must have a monitoring system for safety and efficacy, and they had an independent monitoring committee for all their phase 3 trials. The FDA, a little bit later, issued some monitoring guidelines for trials which come under FDA jurisdiction. I think it's fair to summarize that trials now really must be monitored in a more systematic fashion than probably they were in previous times. If you're monitoring data, especially for those with a monitoring committee, what things are you looking at? Well, you're looking at recruitment because you want to make sure that you're going to achieve the goal. If you don't achieve the goal of patients, you are basically going to have a failed trial. Look at the baseline variables to see whether the patients who are actually being randomized, in fact, are eligible, and are the patients in treatment A comparable to patients in treatment B. You're going to look at outcome measures because you're trying to assess benefits and risks, so you need to understand the benefits, both primary and secondary, and, of course, the toxicities. You want some assessment of compliance to see are we doing the job we wanted to, and we may look at specific high-risk subgroups. So by and large, what a monitoring process looks at is all the things that we consider important in the trial. You can do a few things. You can decide that the trial should continue with no problems and nothing's modified, continue as planned. You may recommend some modification, such as loosen up the eligibility criteria, or drop the frequency of certain measurements which are problematic, or maybe you recommend terminating the trial. You could recommend terminating the trial for a few reasons. One is that you've identified an unexpected and serious toxicity. Toxicity is unacceptable relative to the benefit. You may have such overwhelming benefit that it's considered unethical to continue. You've already established, beyond reasonable doubt, that the treatment is beneficial. You may also decide to stop the trial because the way things are going, it has no chance. There's no trend of interest. It's not really significant. And because of the cost or the risk, if it has no chance of benefit, then this is a trial that should not be continued. Once in a while, hopefully not often, there's a design flaw that's just too logistically difficult to fix. And so stopping and starting over may be the wisest decision. The process of stopping a study, though, is complicated. You need to understand that as you read studies in the literature that have been terminated, or you're participating in it. There's all the primary and secondary outcomes. There's the safety issues. There's both internal and external consistency. There's that benefit risk. There's always a sensitivity to the patients in the trial, the current patients, versus the impact on future patients who might come in the trial or might be treated. There's a clinical versus public impact. If you don't convince or change people's mind about benefits or risks of intervention, then you haven't achieved much. And then there's a list of statistical issues. So we've stated that for ethical, scientific, and financial reasons, there's a mandate to monitor trials for safety. However, the process of repeatedly testing, accumulating data, causes a statistical problem, called the repeated testing problem. Let me illustrate. Coronary Drug Project, which I alluded to earlier, was one of the earliest trials looking at ways to lower cholesterol levels in men who'd had a heart attack. One of the cholesterol-lowering drugs, clofebrate, was tested versus a placebo arm. There are other arms which I won't discuss now. But in this Kaplan-Meier graph of mortality, you can see that the two curves are essentially the same. Over the 7 to 8 years of the trial, there was no difference in the mortality rates from the clofebrate arm versus placebo. However, if you were to have plotted the data as it came in to the statistical center in real time, in calendar time, and every now and then did a statistical test of clofebrate rates versus placebo rates, you would have seen a different picture. On this graph, I plotted on the x-axis months in the study from 0 to 100, which was sort of the end of the study, and on the y-axis the z-value, the normalized test statistic. A z-value of +2 or -2 would be equivalent to a p-value of 0.05. So if one were to apply somewhat naively the standard statistical tests and p-values and look at them in this particular study, there were on the lower half of the graph 1, 2, 3, 4 times, 4 circles, when the statistical test was exceeded or almost approaching a value of -2.0 or a p-value equivalent of 0.05 two-sided. So if you had been somewhat naive in this testing process, one could have claimed on 3 or 4 occasions that the drug was superior to placebo. And yet, if you go back to the previous slide, you can see that in the end, there was actually no difference in mortality. Well, this is an example of the repeated testing problem. And so when you're monitoring trials, which we ethically must do, and scientifically also need to do, one has to be aware of this problem. We could have at least 1 or 2 lectures on the issue of how one interprets interim results, and among them are something called sequential boundaries. Let me give sort of a simple explanation of this graph. We've assumed in this trial that we're going to look at the data 5 times. 1, 2, 3, 4, 5 on the x-axis. 5 interim looks at the data. And again on the y-axis I had the standardized normalized statistic. So instead of plotting +2 and -2 as the naive boundaries, I plotted some other boundaries. They're labeled the Pocock boundary, the O'Brien-Fleming boundary, and the Peto boundary. Let's just pick the Pocock boundary first. It's the open circled horizontal line. And what the Pocock boundary suggests is that in order to protect ourselves and not increase our chance of a false positive, we will demand stronger evidence, larger z-values. In this case, if you were to have the exact values, it's 2.4. So it says that for the z-value to be judged interesting, i.e. significant at the 5% level, the intermediate results, in fact the final results, would have to be a value at least 2.4 or higher. But there are other plans. There's one labeled O'Brien-Fleming, which is the closed circles, and it starts out very conservative, very high in the positive or negative direction, and comes down slowly and approaches a value at the end which is very close to +2 or -2. This boundary also controls for the false positive rate at 5%. So now we have 2 plans that each give an overall false positive rate of 5%, but in a different way. And there's a third graph, the open triangles labeled the Peto boundary. This plan demands even stronger evidence for early termination, value of +3 or -3 or greater in all intermediate results. And then the final result, because we've been so cautious, so conservative, my final result could be a value of almost +2 or -2, the conventional naive value. Well, all these boundaries achieve the same thing, a false positive rate of 5%, but they achieve it in a different way. And there are many more such solutions to choose from. Probably the 3 that are depicted here are the 3 most commonly used ones. In fact, the Peto and the O'Brien-Fleming are probably the most commonly used ways of interpreting interim results. So one don't react too quickly and wind up as they might have been in the Coronary Drug Project, claiming a result to be significant, and in fact it was not. At any rate, this is a very important area that you need to pay attention to as you read studies or plan your own. This idea was applied in the beta blocker heart attack trial, in a trial that was completed in 1981. This is the mortality curves in a population of following a heart attack, half of whom were given propranolol, a beta blocker, the other half placebo. The z-value for this, comparing those 2 curves, the standardized statistic of 2.82. If you were to have plotted those results over time using the O'Brien-Fleming boundary, This graph shows months into the study from June of 1978 to June of 1982. You can see that the results, the dark circles, closed circles, start out with a first z-value of 1.58, got larger, past a nominal naive value of 1.96 or p-value of 0.05. Because of the sequential boundaries in place, we continue to follow the patients until they cross our O'Brien-Fleming boundaries. There's many more other complications that were considered, but for now, crossing that boundary certainly triggered a discussion which led to the recommendation of early termination. Now finally, I want to examine an issue that says what data should be analyzed. This is a challenge that many people sometimes overlook. We could have conducted the best trial, designed the best trial, conducted it in the very best way, and we get all the way to the end and begin to analyze the data. We can introduce more bias than perhaps we avoided in all the design and conduct efforts. There's a principle here which is called the intention to treat principle. It says you need to analyze all patients that are randomized. There is no escape. If you're randomized into the trial, you must be accounted for. There are many, as I say, lookalikes, people who claim they're doing intention to treat, but in fact, they've allowed patients to be excluded, taken out of the analysis. By exclusions here now, I'm not talking about the patients who were screened but not randomized, because that has an impact on which you can generalize to, but the validity of the trial is still okay. But if you start withdrawing patients that you've randomized, my argument is that you can introduce a serious amount of bias. Let me illustrate a couple of simple examples. One rationale for taking patients out of analysis is the fact, well, they weren't eligible to begin with. After you randomize patients in the study, downstream, you discover that some patients did not in fact meet the entry criteria. You got the lab results back, you got the EKG back, you got the lung scan back, and you realize that they didn't have exactly the disease that you thought they had. And your concern is that if you allow these ineligible, strictly ineligible patients to be in the analysis, you may dilute results and miss effects. Remember, we talked about adjusting our design to account for this dilution. The temptation is to take these patients out. After all, they were not eligible. But my point will be that this being done after the fact can lead to bias. Let me share with you an example which was very influential in how academics and regulatory agencies viewed this issue. The Antran reinfarction trial was a trial conducted in the late 1970s and published in 1980 in the New England Journal of Medicine. It was randomized, double-blind, placebo-controlled, studying Antran, a platelet-active drug, versus placebo in patients who had recent heart attack. There were 1,629 patients randomized, and after the fact, after the trial had been done, re-examining the data, 71 of those 1,629 patients were considered to be ineligible. 38 in the control arm and the treatment arm, and 33 in the placebo arm. Well, 38 versus 33, that's roughly speaking the same in each group. The reasons for being ineligible, well, about a third of them, their MI was the patient taking place too long ago or not long enough ago. In some cases, about a third, the MI or alleged MI was not severe enough. The enzymes that sometimes are used to define an MI were not elevated sufficiently. Or there were other issues, competing issues, that sort of made the patient not an ideal candidate for the trial. So the rationale for ineligibility seems reasonable, and the number is about the same. Why not take them out? Well, the next slide here illustrates results looking at the patients who were eligible, ineligible, and the total. We're looking at mortality results here. So the top line accounts for all patients in the study and all known deaths. 74 in Antran, 89 deaths in placebo, definitely reduction, but not meeting our conventional criteria for significance. P-value is 0.20. Now, if you were to examine just the eligible, those who the investigators after the fact believed were the ones we should focus on, there's 64 deaths versus 85 deaths in placebo, an event rate of 8.3% versus 10.9. The p-value for that is 0.07, so it's not quite significant, marginally significant, you might say. In the earlier results that were presented, that p-value happened to be slightly less than 0.07. It became non-significant with the cleanup of the data. But here's the really important line. Look at the ineligible patient, the 38 and 33. In the 38 patients who were in the Antran arm and declared ineligible, there were 10 deaths. Of the 33 patients, in the placebo arm there were 4 deaths. 10 versus 4. Well, that difference is not significant because it's very small numbers. One may not expect maybe to be significant. What I want to point out to you is the event rates compared in eligible versus eligibles. In the placebo arm, it's 10.9% versus 12.1. Roughly the same, p-value 0.92. But notice that comparison in the treatment arm, the Antran arm. It's 26.3% in eligible patients versus 8.3 in the eligible. A difference that's highly significant, p-value of 0.0001. So, the impact of these ineligible patients is non-trivial. In fact, when one looks at the data, it's hard to imagine that some bias, somehow, did not creep in to the post hoc assessment. This has been discussed in more detail in a note by Bob Temple and Gordon Pledger in the New England Journal of Medicine in 1980. As a result of this trial and these results, the intention to treat principle became the principle for the FDA and many academic researchers for demanding that all patients be accounted for, i.e. we use the intention to treat principle, which says you must account for all patients in all events. Another commonly used reason for withdrawing patients from the analysis is that they did not comply. This argument was put forth in papers in 1979 and 1980 in the New England Journal. And 2 types of trial were discussed. The intention to treat a principle, which is management trial, as you intend to treat, but the patients may not exactly do as you intended. But we're going to compare all subjects regardless of compliance. Alternative to that is the explanatory or the per protocol trial, where you try to estimate the optimal effect, understand the mechanism, and analyze only subjects who fully comply. This sounds quite compelling and logical. And when you think that the withdrawals will lead for the non-compliance may lead to bias and dilute your results. Let's look at an example of why this might be not a good idea. This example comes from Carol Redmond and her colleagues in a cancer study published in Cancer Treatment Report in 1983. Here we're looking at a breast cancer study with adjuvant therapy. We're looking at disease-free survival, that is alive without recurrence of the disease. And the time scale is years post-mastectomy. We see 3 graphs here based on degree of compliance. The top graph, the closed circles, are those patients who took 85% or more of the drug they were assigned. The open triangles are those who took 65 to 84% of the therapy drug they were assigned. And the open circles are the poorer compliers, those who took less than 65%. I want to point out to you the method that was used to define compliance. It was total dose received divided by total dose one should have gotten according to the protocol. Well, you can see here that results are just as one might expect. If you take the therapy, the assigned therapy, that you get good results if you're a good complier, mediocre results if you're a mediocre complier, and poor results if you're a poor complier. So far, this all fits very nicely. But suppose, next slide here, I change the definition of compliance, the method by which I define compliance, just a little bit. Same setup, a disease-free survival versus years following mastectomy. But notice I have a second method for defining compliance. The numerator is the same, total dose received. But the denominator is different. It's the dose that you should have gotten while you were still on the trial, while you were still coming in to your clinic visits and so forth. A reasonable definition, I would say, but notice the impact that that slight modification in definition has on results. In this case, the top graph is the open circles, which are the poorest compliers. That is, the poorest compliers by this definition do the best. The closed circles in the middle, which are the best compliers, and the mediocre compliers have the worst results. We've reordered the results by just a slight adjustment in the definition of compliance. So there's something about compliance that's tricky. The second message for this is that the arm that we're looking at here is not the active therapy, but the placebo arm. This arm we're looking at, broken into good compliers, mediocre compliers, and poor compliers, are looking at compliance to placebo. This is a powerful message which you need to retain, that compliance is an outcome. And as soon as you start subgrouping your analysis by compliance, you can virtually get any result, any ordering that you want, even if you're studying placebo. So, for that reason, and there are many other such examples in the literature, we insist that you not have your primary results based on the compliers or the per protocol, but the intention to treat all patients randomized, all events observed. So to summarize these fundamentals, I would say that the randomized controlled trial, as I briefly described, remains to be the gold standard. It minimizes the biases that we can identify and controls for the biases we can't measure. However, we must discipline ourselves to define the question, the outcome measures, and the populations carefully in advance. We must design our trials to have adequate power so we can detect the differences we're looking for, or otherwise one might argue the trial is not ethical, it's certainly not a good financial investment, and it'd be scientifically disappointing. Which design you pick of the many choices really depends on the question that you're asking. You must have a monitoring plan. It might be a simple plan, or it might be as elaborate as requiring a separate independent monitoring committee. But in either case, a monitoring plan is mandatory. And finally, one must be careful not to introduce bias in analysis after you've worked so hard for so long to design and conduct and implement a high-quality, unbiased trial. If you follow some of these principles in your reading and your design of studies, you will have far better research results and more credible results than if you do not follow this principle. Today we've briefly summarized these principles. Each of these principles in themselves are worth a much more in-depth discussion, but I think I've tried to give you an overview that would hopefully help you a great deal in thinking about studies and certainly in reviewing and reading the literature. We talk these days about training our researchers to be better informed. Sessions such as this are at least one step in the right direction. I would encourage you to attend workshops to get more detail. For those of you who are interested in a career in clinical research, there are now emerging programs which will offer you a certificate degree and give you some credentials. If you really are interested in an academic career, perhaps one should even consider a master's degree, or if you really want to push the frontier of research, getting a PhD degree. But this training pyramid is now part of the NIH thinking in terms of how we should train our young investigators, our new investigators, the next generation, in being prepared for careers in clinical research. And even if one is just a practitioner using these results, some of this training would still be quite useful. At any rate, I have been very happy to have been able to share this time with you, and hopefully at some point we may be able to speak directly either by phone or through the email. Thank you very much for your time, and best of luck in your reading and in your research careers. Much of the material that we've covered today in these three parts can be found in a textbook that I co-authored with two colleagues of mine, Dr. Lawrence Friedman and Dr. Kurt Furberg. This book, called Fundamentals of Clinical Trials, has been around for about 25 years now, going on 30, and it has served well in sort of introducing you to the concepts and the fundamentals of clinical trials. If there's topics that we didn't have time to cover today that you're interested in, I would encourage you to take a look at some of the chapters in this textbook for further information. Thank you very much.